തളത്തിന് എടുക്കலையா ഓടി ഓഫ് ഉണ്ടോ பாத்தீங்க ഹലോ ഹലോ
Right. Good evening, people. Good evening. How are you? Can you hear me clearly? Please let me know that. Can you hear me clearly? I know other. Right. You can. So today, people, we are going to have a quick discussion on plant growth and development, second growth, ne? Plant secondary growth. Can you hear me clearly? Please send us a small message. Someone? What message dan lamai? Yes or no? Right? Yes, Loharani. Okay, good. Perfect. All right. Now, today's plan, people, will quickly go through the second growth part first, right? Second growth. And then we'll talk about the cell structure and function part is there, and animal diversity part is there. Okay. So, secondary growth, plant secondary growth. Now, people, secondary growth of plants. We learn secondary growth is happening in some of the plants, not in all plants. We said people secondary growth is present in all gymnosperms. It happens. Yes. Slow have a Right. Right. People plant secondary growth. Yes, yes. How is that? Yeah, okay. All right. So, secondary growth, people will quickly go through secondary growth. Yeah, right. Primary growth, secondary growth. Right. Second growth in plants. Second growth, increasing in the diameter of the stems and the roots in plants due to the new cells produced by lateral meristems. Now, what are lateral meristems, people? Lateral meristems, you learn, people, apical meristems available and lateral meristems present, right? So, apical meristems and lateral. So, apical meristem, root apex, and then the shoot apex as apical meristem. Lateral meristems are the meristems present laterally in the plant body. Meristems present laterally in the plant body, right? So, you learn, people, these classification of meristems, we classify meristems. We classify meristems. We said that, people, the meristems at the tip of the shoot, meristems at the tip of the root, they are the apical meristems. And then lateral meristems are present laterally in the plant body. So, there are two lateral meristems in the stem. So, if I show you the level, the border between the stem and the root, we can show the border like this. This is the border. So, then we can see, people, this is the vascular cambium. This is vascular cambium, a secondary dividing tissue. And then this is the next cylinder. Can you tell me what that is, people? This is the second, the lateral meristem present in the stem. What is that? Second lateral meristem present in the stem. What is this one? So, this is the vascular cambium. This is vascular cambium. Vascular cambium of stem. And this one we call, can anyone tell me what that is? Come on. Can someone tell me what this is? No one is going to tell that. Okay, so that's bad. So what is this one, people? This is we call as the coke cambium of stem. Now, same like that in the root. Same like that in the root, you can see, people, two dividing tissues present. Same like that in the root we can see people. Neither. The lateral meristems are there. One of the lateral meristem is this one. This is the vascular cambium of the root. And this one, people, I think some of you know that. What is that, people? Yes, cocambium of the root. Very good. This is cocambium of the root. I have a good student. How the Saturshana. Right. So, this is the people, cocambium, cocambium, cocambium of root. Right. And then this is vascular cambium, vascular cambium, vascular cambium of root. So everything together, all these, what do you call, people? Lateral meristems. Lateral meristems. Lateral meristems. All of them together, what do you call? Lateral meristems. Now, people, you learn the apical meristem, shoot apical meristem, root apical meristem. You learn lateral meristems. There is one more meristem type. Only the good students can tell me the other meristem type. Who is going to be the good student here? Right? There is another type of meristems available here. What is that meristem? Yes, very good. Lavanya says intercalary meristem. That is correct. So, can you tell me whether the intercalary meristem is a primary meristem or secondary meristem? Now you learn a color code. We put people red color. In the class, we put red color people for the primary meristems. So, I'm going to change the color now little bit. Red color here. Red color. Primary meristems are red in color. Secondary meristems are blue in color, right? And then you know where intercalary meristems present? Intercalary meristems are present, people, at the base of the leaf. Now, not normal leaves. We said that people in the monocot plants at the base of the leaves and between the nodes, we can see that the intercalary meristem present. Now, your question is whether the intercalary meristem is primary or secondary. Intercalary meristem, primary or secondary? Can you answer that question, people? Intercalary meristem, whether it is a primary or a secondary one. Answer my question, right? Answer the question, right? Primary or secondary? Who's going to answer? Intercalary meristem, primary or secondary? Come on, come on, come on, come on. Yes, intercalary meristem. I think I have to change the color here. If I'm putting a color, then people, my color is going to be red. So that means, people, intercalary meristem is a primary meristem. It is primary meristem, right? Now, you learn, people, the secondary growth process is happening in all gymnosperms. The resource book says, right? Resource book says, people, right? Resource book says increasing in diameter of the stem and roots due to the new cells produced by lateral meristem is called as a secondary growth, right? And this occurs in stems and roots of woody perennial plants. Woody perennial plants. What do you call perennial? Plant growing many years, you call them as perennial. Woody perennial plants, right? So perennial plants, this happens including all gymnosperm. Look at this, people, including all gymnosperms. Very important word. What is that? It happens in all. It happens in all gymnosperms. All gymnosperm species. That's very important. That is very important. Happening in all gymnosperm species. And then not in, you can't say all angiosperms. And many of the dicot species. Many of the dicot species. Many of dicot species this happen too. Right? Many of the dicot species this happens too. Yeah. Oh. Oh. Right. So I think I have to use this tool, using the wrong tool. Right. Right. Now look at this. Including all and many of the dicot species. Many dicot species, ne? Lateral meristems, namely vascular cambium and cocambium. Look at these people. Namely vascular cambium and the cocambium produce cells and tissues in the secondary growth. Produce cells and tissues in the secondary growth. And then the vascular cambium adds vascular cambium. The vascular cambium adds secondary xylem towards primary xylem and secondary phloem towards the primary phloem. So to clear that, people, let's check the diagram, right? Let's check this diagram. Today my setup is not perfect, right? We have just people arrange this to discuss, maintain, or to begin the discussion, right? But I'll set it up fully later. Now, people, if you think of the secondary growth process, you learn secondary growth. This is the primary stem, neither. This is the primary stem. So these are the vascular bundles. These are vascular bundles of the primary stem. Vascular bundles of the primary stems. So you have to draw, people, at least 6 vascular bundles, neither. So this is the primary grown stem. So let me show that in 3 dimension. This is the primary grown stem. And then the people here, we can see these are vascular bundles. Xylem and phloem present like bundles, neither, vascular bundles. Then people, in this vascular bundle, you can see some dividing cells present. So you can call them as cambium. These are dividing cell layer present inside. Cambium. You can say cambium, intravascular cambium, or else you can just say cambium. A cambium present inside. Then people, red color part. Can you tell me what this red color tissue is? This red color tissue, what is it? Red color tissue present. What is this red color tissue? This one is the primary xylem. This is primary xylem. Then I'm taking, people, green color. Hope you can see green color, right? The part that I color now, that's the primary phloem, neither. Primary phloem. So what happened, people? During the activation of the growth, secondary growth process, first of all, a ring of dividing cells are going to form by interconnecting these cambium with some of the parenchyma in between, people. This is now forming a ring of dividing tissues. So let me show that. This forms a ring of dividing tissues like this. A ring of dividing tissues. This ring of dividing tissues we call as the vascular cambium. This ring of cells we call as a vascular cambium. So when the vascular cambium form, from the vascular cambium inside, what do you have, people? From the vascular cambium inside, what do you have? What do you have? Primary xylem. From the vascular cambium outside, what do you have? You have the primary phloem, neither. So once this form, the vascular cambium is going to form, people, towards inside, right? Vascular cambium is going to form inside the secondary xylem. Vascular cambium is going to form towards outside the secondary phloem, right? So I have to draw the next diagram for that, right? Can you take a piece of paper and then draw this diagram with me, right? Can you draw this second diagram with me, right? Vascular cambium adds secondary xylem towards primary xylem. Vascular cambium is adding secondary xylem towards the primary xylem, right? And primary xylem and secondary phloem towards the primary phloem. So I'm going to draw the next diagram, right? This is my vascular cambium. This is my vascular cambium. This is the vascular cambium. It's not clear, ne? This is the vascular cambium. This is vascular cambium, right? And from there, new cells are going to form towards inside. New cells are going to form towards inside. New cells are forming towards inside, that is the secondary xylem. Inside the secondary xylem forms, and secondary phloem is going to form towards outside. Secondary phloem forms towards outside. Look at this. This is the secondary xylem. This is secondary xylem. So this is simple part. This is secondary xylem. So I know that you have studied these things, but I'm just trying to help you people to revise these sections, right? So this is the secondary phloem. Sorry, secondary xylem. So how about secondary phloem? Produced towards outside. Produced towards outside. So this is the secondary phloem. Secondary phloem forms towards outside. This is the secondary phloem. Right? Secondary phloem. So where is that primary parts? These are the primary xylem. 1, 2, 3, 4, 5, and 6. This is primary xylem. This is primary xylem. So you can see that primary xylem present. Primary xylem. And then people, against the primary xylem, what do you see people? Primary phloem. So this is primary phloem. Against every primary xylem, you can see that the primary phloem present. Right? Primary phloem. Right? So you learn people this vascular cambium. This vascular cambium does several events. What is that, people? Producing xylem towards the primary xylem, producing secondary xylem towards the primary xylem, producing secondary phloem towards the towards the outside. The resource book says, people, it says xylem and secondary phloem towards primary phloem. Secondary phloem towards the primary phloem. That is how they describes it, right? And then this is the outermost tissue layer, epidermis, and the remaining tissues. And then people, this vascular cambium can produce some parenchyma. They are forming something called as a vascular race. So I'm going to show you vascular race like this, right? So when they form, most of the cells, when they involve in forming xylem and phloem, few cells of the vascular cambium are involving in forming parenchyma. So what do you call them? Vascular race. The vascular race. So this is the next stage of the diagram. Now only one thing is missing. What is that? The cocambium. Now what happened? The stem diameter increases. This is tend to rupture, right? Stem diameter is increasing as the internal tissues are growing. Now what happened? New tissue layer comes immediately inner to the epidermis. New dividing cell layer comes. New dividing cell layer forms. What is this dividing cell layer? Can someone tell me what that is? What is this cell layer, my dear? Can you tell me what that is? Come on. What's that new cell layer? Come on, please respond to me. What is that cell layer, people? That's the cocambium. So this is cocambium. So what is formed by the cocambium? From the cocambium towards outside, what it forms, people? Coc cells. So coc cells are going to form towards outside. Right? So let me show you here. Coc cells are going to form towards outside. Inside, people, some cells are formed. You don't have to bother about that. Right? Coc cells are going to form towards outside. We'll take it like that. Right? These are the coc cells. So who forms these coc cells? Cocambium forms coc cells. This is it. Coc is formed towards outside. coke forms towards outside. That's it. This is the secondary growth. Secondary growth, right? And then people, some regions in this coke, the spherical shaped coke cells are produced. Now these coke cells are spherical shaped coke cells are produced, providing a site for the gas exchange. So here and there you can see that these spherical shaped coke cells are present, providing some holes for the effective gas exchange, providing some gaps for the gas exchange process. These points with these spherical shaped coke cells, you see them from outside as horizontal slits. You see them as horizontal slits like that. And these horizontal slits, we call them as what lenticels. They are called as lenticels. Anyway, this outer layer, there are some ruptured epidermis present. Some ruptured epidermis present outside. Some ruptured epidermis can be there. Right? Ruptured epidermis present outside like this. Ruptured epidermis. So you can label this diagram. This is ruptured epidermis. This is ruptured epidermis, this one. This is coke. This is coke cambium. Now, coke and coke cambium together. Do you have any name for that? Coke and coke cambium together. What do you call that? Come on. Coke and coke cambium together. What do you call it? Come on. Come on. Chat with AI companion. Try it now. Right? Right. Coke and coke cambium. Can't you send your messages to the who's this? Main chat host, co-host. Right? So now the Zoom new settings of Zoom is not good because I can't see unless I click that, I can't see the chat. That's my problem. Right? So if you can send it to the main chat. All right. Periderm, very good. Zikra. Correct. Periderm. Coke and coke cambium together is the periderm. So there is a question now. Bark. What is bark? Now, what is bark, All tissues outside the vascular cambium is bark. All tissues outside the vascular cambium. So we can take this part as bark. Bark is what? All tissues outside the vascular cambium is bark. So what tissues included into the bark? Vascular cambium is not part of it. Can you tell me, what tissues included into the bark? If you go from here to this side, you have secondary phloem, you have primary phloem, you have this cortex, you have the coke cambium, and you have the coke and the ruptured epidermis. All are parts of the bark. So the main tissue present in the bark is phloem. Main tissue present in bark is phloem. Main tissue in the bark is phloem. You understand that? Main tissue in the bark is phloem. Right. And then then we have to discuss something called as the annual rings and the growth rings. Right? Annual ring and growth rings. All right, people. Now, this vascular cambium is like you. Its activity is not constant throughout the year. Sometime periods the vascular cambium is fast. Sometime periods the vascular cambium is slow. Right? So therefore, the activity of the vascular cambium can change. Sometimes some periods, the environmental conditions are so favorable. Right? Nice environmental conditions. How about your environmental condition now? Is it good or bad? How is it now? Your surrounding environment? Good condition, no classes. Did you go to school today? Did you go to school? So you went to school, came home, and then relaxing afternoon. And now you're peacefully listening to me. Right? You have to do the paper. Right? So the coke, vascular cambium activity changes from time to time. Some periods vascular cambium activity is optimum. It produces more xylem. Some periods it's produce people less xylem. So depending on that, we can see people, the amount of xylem produced can be variable. Some bands are going to appear. When this vascular cambium works faster, more tissues are added, light color tissues are added. Ne? And then some periods when the vascular cambium activity is less, then they are producing more dark color tissues. So you know people in some countries, the climatic changes are seasonal. So some periods are better, some periods are bad. Neither. So depending on that, the action of the vascular cambium can change. Action of vascular cambium can alter. So what I'm going to do now, I'm going to show this more clearer by enlarging this diagram. Look at this. This is the vascular cambium. I'll draw it little wider. This is the xylem, primary xylem. This is primary xylem. This whole part is the secondary xylem now. This whole thing is the secondary xylem. Ne? This entire part is the secondary xylem. This entire region is secondary xylem. And here we have the primary xylem. Here we have the primary xylem. And then you know people relatively less phloem is produced compared to xylem. So this is secondary phloem. This is secondary phloem. This is secondary phloem. Secondary phloem. Secondary phloem. So we can show you the primary phloem. We can show you primary phloem against the primary xylem, primary phloem. Against this one, primary phloem. Against this one, primary phloem. Against this, primary phloem. Against this, primary phloem. Against this one, primary phloem. Right? So we'll color it little bit. Let's color it little bit. So this is the arrangement. This is the arrangement. And then this is the arrangement. Right? Secondary xylem. Then here people, this is the cocambium. This is cocambium lamai. This is cocambium. And the cook. And cook. This is cook. Right? That is cook. I'll take it as 2 layers. Cook. This is cook. That's cook. Right? Now, something is missing. Ne? What are those people? These vascular rays are missing. Vascular rays are missing. Ne? So we draw it like that. Vascular rays. Right. Now, we are to discuss people the rings, growth rings and annual rings. Yeah, growth rings and annual rings. Now, some periods, this vascular cambium, it had produced people dark color xylem. Dark color xylem, ring of xylem dark color. They are actually formed during the unfavorable periods. Now, I'm showing you people 2 dark color rings now. So, they are 2 unfavorable periods. Right? So, this is another unfavorable time period. This one. unfavorable time. This one also unfavorable time. So what do you have in between now? Favorable, nice time period, this one, right? So therefore we can see that set of rings are going to appear like this due to the variation of the activity of the vascular cambium, right? Due to the variation of the activity of the vascular cambium, these rings are going to appear, right? So what do you call, people? What do you call? The dark ring and the lighter ring, right? Lighter ring occurs, people, during the favorable time. So what do you call that wood, people? Favorable time period wood. What do you call it? Favorable time period wood. What do you call that? Favorable time wood. Huh? Come on, type it quickly. Favorable time period wood is the, come on. Favorable time period wood, what do you call it? That is the spring wood, ne? That is spring wood. Spring wood. And then the unfavorable time period wood, people, what do you call that? The summer wood. Summer wood. Spring wood and the summer wood. Now, together, what do you call these two, people? These two together, what do you call? Annual ring. Annual ring. Annual ring occur in temperate region. Now in Sri Lanka, tropical region, we don't have spring and summer, but we have favorable, unfavorable. Then we don't call them as, we can see similar rings which are not corresponding to 1 year growth. Now here, spring wood and the summer wood together corresponds to 1 year growth, right? Now in Sri Lanka, we have favorable, unfavorable, couple of favorable, unfavorable throughout the year. So therefore, they are not corresponding to the 1 year growth, the rings that appear, light and the dark ring. So we call them as what, people? They are called as growth rings. So the annual ring and the growth ring, small difference is there, ne? Right? One more thing. What is that, people? Very recently formed a xylem. Recently formed xylem is the xylem present closer to the vascular cambium, right? These are actually the early formed xylem. Now, this is the recently formed xylem closer to the vascular cambium. These regions that I am darkened now, they are the early formed xylem. They are the early formed xylem. These early formed xylem, we call as the, what do you call it? Heartwood. Because they are the wood present at the center, the heartwood. Wood present closer to outside, what do you call that as the sapwood, heartwood and the sapwood, right? So heartwood and the sapwood. So we can see a difference between these two, heartwood and the sapwood. So I'm showing you a region, people. I'm trying to darken it, right? And then this relatively light color xylem closer to the vascular cambium is the sapwood, right? That's the sapwood. Look at this. It takes some time for me to put dots all over, right? I don't think it's necessary. The point is now clear for you, neither. I think this is clear for you. Clear for you, right? Okay. No need to put the dots, ne? Completely. So let's quickly go through the note now. Let's go through the note. Let's go through the note. Right. Right, now let's go through the note quickly. The vascular cambium add a co-cambium that we said, ne? The vascular cambium add the secondary xylem towards the primary xylem and secondary phloem towards the primary phloem. The co-cambium produce tough, thick covering. The co-cambium produce tough, thick covering consisting mainly of vaccine-pregnant cells, protects the stem from water loss and from invasions of insects and bacteria and fungi. The woody plants, in woody plants, primary growth and the secondary growth occur simultaneously as the primary growth add new cells, lengthens the stem and the root. In the young region of the plant, secondary growth increases the diameter of the stem, roots in older regions. Okay, let's move to next page. Secondary vascular tissues are produced by the action of vascular cambium. Let me share this people for you. Okay, now it's clear. Ne? Now it's clear. Secondary vascular tissue is produced by the action of vascular cambium. In a typical body stem, the vascular cambium consists of continuous cylinder of undifferentiated cells of often only a single cell layer in thickness. So that's a very important point. What is that? Vascular cambium is always a single cell layer, right? Vascular cambium is only a single layer in thickness located outside of the pith and the primary xylem and to inside of the cortex and the primary phloem. So this position of the vascular cambium, they mention in a special way. Ne? So how they say, people? Located outside the pith and the primary xylem and inside of the cortex and the primary phloem. So it's sandwiched between the primary xylem and primary xylem and primary phloem. In typical woody root, the vascular cambium forms laterally exterior to the primary xylem and interior to the primary phloem and pericycle. And as these meristematic cells divide, they increase the circumference of the vascular cambium and also add secondary xylem. Viewed in a cross section, vascular cambium appears as a ring of initial. This is important. Some initials are elongated. Some initials are elongated and are oriented with their long axis parallel to the axis of the stem or root. Right? These elongated initials are producing people xylem and phloem. So these tracheids, vessel elements, parenchyma, fibers, neither of the xylem as well as seed tube elements, companion cells, phloem fibers. So they are making the xylem and phloem. The other initials are shorter. Look at that, people. Shorter and oriented perpendicular to the axis of the stem. They produce vascular rays, mostly parenchyma cells, right? At the secondary growth, as the secondary growth continues over many years, xylem would accumulate. The wall of the secondary xylem cells are heavily lignified, blah, blah. During early stages of the secondary growth, the epidermis push outward, causing it to split. So, a cylinder of dividing cells that arise in the outer layer, and this is becoming meristematic and form what, people? Co-cambium. So, it forms co-cambium. It forms co-cambium. It disappears suddenly, right? Co-cambium. So, what's wrong with that? No, it's okay. Ne? Co-cambium. So, people, this story is simple. Ne? How the tissues are formed, people, right, is described here. So, co-cambium is produced. Ne? coke cambium is produced? Right. Look at this. Coke cambium produce coke cells to the exterior. Coke cambium produce coke cells to the exterior. Right. To the exterior and to the interior. Cells addressed to the exterior become the coke. That's simple. Coke cambium and tissues produce are collectively called the periderm. The coke tissues function as a barrier that helps to protect the stem from water loss, physical damages. Look at these people, how it is protecting water loss, physical damages, and pathogens. So the coke cambium function as a barrier and helps to protect the stem from water loss, physical damages. Each coke cambium and the tissues is produced comprises layer of periderm, which is impermeable to water. Hurry, we'll move to next part. Page, right. For gaseous exchange, small pores are present in the periderm, known as the lenticels. We talked about it, neither. Lenticels are there, which are formed by loosely arranged coke cells. They appear as horizontal slits. Further growth of the stem or the root breaks the layer of coke cambium and it lacks its meristematic activity and it's becoming coke cells. A new coke cambium is initiated inside, which produce a new layer of the periderm. As new cells are added, outer region of the coke will crack and peel off in many tree trunks. So that's clear, neither. So this process is clear for you, right? The secondary growth, right? So I have included people in the paper, right? Some of the questions relate with the secondary growth. Hope people, this part will help you, right, to read through the section, right? So read through that, right? Read through that. And then people, shall we quickly go through the animal diversity part as well? I think we can study the cell structure and the function part. So we'll go through people animal diversity lesson. This time people, I said that I'm going to question you regarding the invertebrates. Invertebrates. Did I give you a short note for animal diversity? No, I didn't. Right. So let me see. Right. So animal diversity part. Right. So you have to study the remaining part, people. I explained the basic things, right? Here, the animal diversity. My questions this time, people, are only based on the invertebrates, right? Questions are only based on the invertebrates. So that is Cnidaria, Platyhelminthes, Annelida, Mollusca, Arthropoda, and Echinodermata, right? The questions are only based on that. So let's quickly go through, people. You learn about the animal classification. I talked about, people, the animal classification, how we classify animals, right? So we don't have to go through that. We'll quickly check, people, each phylum and their features. Each phylum and the features, right? So the first one is Cnidaria, right? Cnidarians. They have, people, given very limited information. You don't have to study a lot. Characteristic features of examples are not necessary. So they start with very easy way. So Hydra, sea anemone, obelia, corals and jellyfish, they come under this. So look at this. These animals are marine, except a few freshwater species. Now look, right, look at this. The Cnidarians. What do you know about Cnidarians? Well, very few information you have to remember regarding Cnidarians. Here, few information you have to remember about Nigerians. Few information about Nigerians. What do you know? Diploblastic. So some members are there. They have a sac-like body, ne? This sort of a body made up of two tissue layers. What do you call those two tissue layers? Ectoderm and the endoderm. So I'll show it like this. Then they don't have a mesoderm, ne? The black color layer is the ectoderm, green color layer is endoderm. What do you call these people? Gastrovascular cavity. This is their mouth, ne? Mouth turned up. This is their mouth. Mouth turned up. Then they are attached to a substratum by the basal plate like region, the bottom part. Basal plate. Right? So, and then people, these are tentacles to capture the capture the prey. You learn people, two body forms are there, ne? There is another body form like this. There is another body form like this. What do you call them? These are zooids. Right? Individuals, you can call them as zooids. So this is the second zooid type. This guy is also having similar arrangement but different body shape, ne? Bell-shaped body. Mouth is therefore turned downward. This is mouth. Mouth turned downwards. So here people, the tentacles. Tentacles. Like this one, they are the tentacles. Tentacles present. Then people in the body, we can see special cell type present. Ne? Special stinging cell type is present. What do you call them? They are called the nematocyst or the nidocyst. Right? Let's check this note little bit. Check this note little bit, people, to check those special information. Right? Majority of them are marine. Simple organization, diploblastic. And then outer layer ectoderm and inner layer of endoderm present. Sandwich between these two layers, mesoglia present. Okay, forgot to mention that. Ne? Mesoglia. The layer present in between, it's a non-cellular layer. Mesoglia present. Simple gastrovascular cavity present. And then this cavity is lined by endoderm. Respire through the body surface. Respire through body surface. That means people, they don't have anything special. They show radial symmetry with two body forms, polyp and medusa. Look at this. Polyp and the medusa. Two body forms are there. And then the polyps are cylindrical forms attached to the substratum. And by the aboral end of the body, right? Aboral end. So, oral means the end towards the end where the mouth present. Aboral means what, people? The region where they have the opposite to the mouth is aboral. Ne? Oral and aboral surface present. Oral and aboral surfaces are there. Now, medusa resembles the flattened mouth down version is medusa. Right. Some Nigerians exist only as polyps, some are existing as medusae. So both forms are there. Reproductive structures as sexually and asexually, reproduce sexually as well as asexually. Asexually by budding. That means tiny buds are going to form. Ne? Tiny buds are going to grow out from the body. Right. Moving to next page. Tentacles are armed with nidocytes for defensing. Nidocytes are cells unique for cnidarians. They contain a capsule-like projection, protrude outwards. So let's check that part, people. Right? So they have some of these cells. What do you call the nidocytes? Ne? Nidocyte is the cell. And then they have a cyst inside. Now this part is the cyst. This part is the cyst. So nidocyte with a cyst inside. What do you call these people? Nidocyst. And this can project outward. So there is a thread-like part neither. There is a thread-like part with the spear like this, people, with some toxin. So what happens, people, when the cell contract, this is going to protrude out. So this, it's going to shoot out like this. It can shoot out. This structure can shoot out. So there is a thread-like part. So they say that nidocytes are unique to cnidarians. They contain nidae. The capsule-like projection protrude outwards. So this is nidae. Nidae, the capsule-like part. Specialized nidae containing stinging thread is called nematocyst. So when there is a stinging thread-like structure present, they say it is the nematocyte. No specialized excretory systems are available in these guys. That's all neither. That's all for these cnidarians. That's all for the cnidarians. Then, people, the next one is flatworms. See, flatworms, people, flat guys. Now, are they diploblastic or triploblastic? How many tissue layers form their body? They have how many tissue layers, people? 3 tissue layers are there. Ne? They have 3 tissue layers. Look at this. Look at this. Phylum Platyhelminthes. So, planaria, tinea, fasciola, tinea. What is that? Tinea, people? Taper. Fasciola, liver fluke. Planaria. Flatworms, commonly known as flatworms. Flea-living, and they are found in marine, freshwater and damp terrestrial habitats. Body is dorso-ventrally flattened. Some have elongated tape-like body. Triploblastic, that means what, people? Body made up of how many germ layers? 3 germ layers are present. Ectoderm, mesoderm and the endoderm. This is important. 3 germ layers. Ectoderm, mesoderm and endoderm. So they have signs of cephalization. What do you call cephalization? Formation of the head. They have some primitive head-like region. So if I ask you, what is a head? If I ask, what is a head? You have head. So what is a head? It is kind of a body region, anterior body region with central nerves, and also, people, with central nerves and concentrated nerves, right, where the lot of ganglia are present and sensory structures present. Usually, people, front end of the body, so that it helps the animal to detect the environment, head-like structure. So what do you say about, people, platyhelminthes? The signs of cephalization present. They have signs of cephalization. Now this is a cnidarian. Cnidarian. So this is the anterior body region. These are eye spots. So this region you can see, people, the central nerves present. Central nerves present. They are passing to the posterior region of the body like that. And then, people, we can see that some of the transverse nerves present like this. Then they have, people, digestive tract. What do you say? Irreversible pharynx, ne? The pharynx which can turn inside out. Irreversible pharynx present. And then, people, their digestive tract, if I show you in red in color, the gastrovascular cavity present, people. They have a gastrovascular cavity, right? Gastrovascular cavity present. Gastrovascular cavity. That means, like in cnidarians, these guys are having a cavity, no anus. So no alimentary canal. Now, what do you call alimentary canal? Mouth and anus both present, it's alimentary canal. These guys are not having alimentary canal, right? Mouth and anus only. So that's the arrangement here. Now, people, we learn that special cell type is available for excretion. tiny cell type is available. They attach with the system of tubes like this, right? These are attached to a system of tubes like this. And finally, we can see that some bulb-like cells present. Some bulb-like cells present. What do you call them? These are the flame cells. These are flame cells. Flame cells can collect waste materials and pump them into the track, this tube system, right? So therefore, there are some simple excretory structures available in these animals. Simple excretory structures present in these animals, right? Look at these people. No body cavity, no circulatory, no respiratory, and no skeletal systems. The gases exchanged by simple diffusion. Sensory organs are found only in free living example, eye spots available. First appearance of little complex nervous system and sensory system. First appearance of, this is important, eh? First appearance of little complex nervous system and sensory system. Pair of anterior ganglia present. Anterior ganglia, I showed that, ne? Anterior ganglia. Right? These are anterior ganglia people. So what they say that they have a pair of anterior ganglia and two longitudinal nerve codes. The blue color thing, the elongated one, they are the longitudinal nerve codes present, right? And then check this, appearance of separate organs for excretion, separate organs for excretion. Nitrogenous excretory system consists of protonephridia. Highlight that people, protonephridia, special tube-like system. They are attached to the flame bulb used to maintain osmotic balance too. Incomplete digestive system. Some have reversible pharynx. These they have cilia for locomotion. This is important. I forgot to mention that. Free living forms are having cilia for locomotion. And then asexual reproduction by regeneration. Bisexual but shows cross-fertilization. Now these animals, people, we can see that these animals are bisexual. All of them are bisexual. None of them are unisexual, right? So platyhelminthes, that's like a special feature, right? Bisexual members present. Now, you know, when we discuss about Nidarians, it's not like that. Ne? Some are unisexual, some are bisexual, but these guys are all bisexual. Ne? So that's what you need to know. So some are parasitic, they say. Parasitic forms are present. Let's check the last few points, right? Parasitic forms are there. They have several larva stage. Direct development occur in free examples. Look at that, people. Direct development occur in free living examples. That means they don't have larvae stages. So who is free living? Who is free living out of these? Planaria is a free living one. Eh? That's all. Now, if you take people, liver fluke is not a free living, it's a parasitic one. And then people, if you take the tapeworm is also not free living, right? So the free living ones, they develop without larvae. The other parasitic forms, they have larvae stages. That's a special feature, right? Look at this. In parasitic forms, there are several larvae stages. Direct development in free living stages. That's important, right? Next, we will be here to discuss regarding nematodes. Next, we are to discuss regarding nematoda. Nematodes. Oops. Nematoda. Phylum nematodes. Right, nematodes. What do you know, people, about nematodes? Lamai, nematodes, triple blastic, ne? Triple blastic animals, right? Triple blastic animals. So we learn, people, they were the first animals to have complete digestive tract. Body is having tapering ends like this. Tapering ends are there. Complete GI tract present, ne? Complete GI tract present. Anus and mouth present. That's like a special feature, ne? Anus and mouth present. Right? So they have a alimentary canal. Then what do you said, people? Triple blastic, ne? There was a special point mentioned. They have only longitudinal muscles present in their body. Now, if you ask the muscle arrangement, right, in the plateal method, they have not mentioned it. So you don't have to bother about it, right? But they have both circular, longitudinal, both muscle types are present when you take the plateal mintes, which is not mentioned in the resource book. But here, people, they say only longitudinal muscles available in the nematoda. So it can move in one direction. Eh? Very simple, this sort of movements they show, right? So mouth and anus present. This is mouth. This is mouth. And there is a anus present. This is anus, right? Then people, they have a body cavity. Ne? Inside they have a fluid-filled space. We call it as a pseudocoelom. Body cavity present. Thick cuticle present. Ne? They have a thick cuticle lamai. So that they can live in harsh conditions, so they can resist people. Sometimes, ne? Thick cuticle present. They have some sensory structures. The anterior end of the body has, people, some structures. Papillae. These are some sensory structures present in anterior region of the body. Papillae present, right? And then people excretory structures, some tubes are available for excretion. Unisexual members, right? Let's quickly go through, people, the resource book to see the points. So the examples: roundworms, hookworms, and pinworms. Most of them are free living and live in marine environment, freshwater, damp soil. They are bilateral symmetrical, triple blastic with pseudocoelom. Their body forms are cylindrical with tapered ends. Body size varies from microscopic to macroscopic. So tiny microscopic ones are there. That's a special point, people. Some are microorganisms. These are animals, ne? So animal microbes are there, ne? So these things are important, people, because when later, when they give you questions in the exam, right, these tiny points they pick and ask questions. They do not show distinct cephalization and segmentation. The sensory papillae are present. Now look at these, people. Sensory papillae are present. Sensory papillae are present in the anterior end. No circulatory, respiratory, body wall composed only of longitudinal muscles are very important. Longitudinal muscles present. Longitudinal muscles present. Right? Alimentary canal present. Mouth and anus there, ne? So longitudinal muscles are helping in these animals to locomote. Sexual reproduction is by internal fertilization. That's important point, people. Internal fertilization. Sexes are separated. Females are larger than the males. Ugly. Large females present. Females are larger, fat females. Males are cute. Cute male. Right? This is a male. Right? Sweet male. And then large females. So size wise, we can see a clear difference present. No developed excretory structure. Some ducts are available. They say that, people, some tubes are present, right? But no well developed excretory structures. right? So check that. Oops. Okay, this is stucking a little bit, right? So that's about nematoda. Then come to people, the annelids. Then come to annelids, right? Look at these annelids: marine, freshwater, or live in dam soil. Segmented worms. That's a special feature, Lamai. Segmented. So nematodes over. Now come annelids. So how annelids are different to nematodes? One thing is, people, they are segmented. You know, segments are present to improve the efficiency of locomotion. These animals are segmented. Segments present. We'll take the animal like this. Right? Segments present. Segmented animals. Segmented. Partition present, ne? Partition present. Now these animals are having alimentary canal. Complete alimentary canal present. Now they have alimentary canal. Need not to say complete, ne? Alimentary canal is continuous passage. Right? So if you take a section in the body, both longitudinal and circular muscles present. So that is now variable too, ne? Both longitudinal and circular muscles present. Right. They have people nervous system with some anterior ganglia and segmental ganglia present like this. Segmental ganglia. Now they use a special word, ne? Circummenteric connectives present. Now here around the esophagus, these nerves are passing. So what they say, people? Circummenteric connectives present. And then these are connected by ventral nerve cord like this. Excretory structures present, ne? Specialized respiratory structures, some gills available, right, in these members. So some special features. When come to Annelida, we can say some special features, name people. First animals to have what? The coelom, real body cavity coelom, ne? First animals to have circulation. First animals to have respiratory structures. First animals to have segmentation, right? So they are having a lot of medals, right? Like you, right? They have won a couple of medals in evolution. So this is actually a turning point in evolution, right? They are actually some animals that have done some massive change in evolution. Now, people, these guys are having the locomotory structures like this. They have locomotory structures. So, first to develop people specialized locomotory structures too. What do you call these specialized locomotory structures? Parapodia and setae. Some bristles are there, ne? Some bristles are present too. These bristles are called as setae. Bristles present. Sometimes they can be used for defense, but basically, people, there are some structures to paddle and to crawl to get the grip. Right? Parapodia and setae present. So let's check some details, people. The first animals to show cephalization, nervous system well developed, right? Dorsal cerebral ganglia present. They have people circummenteric connectives that you know now, ne? Circummenteric connectives, very clear, ne? Circummenteric connectives, right? dorsal cerebral ganglion, ventral nerve cord present. I discussed them. Then clitellum present. That's a special feature. Clitellum available, right? Clitellum present. Clitellum. What is clitellum? Here people, in their anterior segments are forming slime. And these slimy casing, therefore they can produce. These anterior body segments are the segments with the genital opening. So what you call the sex organs are located in these anterior segments. So what they do, they release sperms and eggs. They release sperms and eggs. These anterior segments are releasing sperms and eggs. So these animals can be bisexual. Some are bisexual, some are unisexual. So they release sperms and eggs. And these sperms and eggs, they're going to fertilize people in this slime released by these segments. So these slime which is released by the segments are now helping in secreting a cocoon. Right? So the clitellum secretes a cocoon. Clitellum is this glandular segments. Clitellum is the glandular segments. Right? Clitellum is glandular segments. People, are you with me? Are you still with me? I can't see the participant number. I just want to check. I don't know whether I'm like talking alone, like crazy. Are you with me? I can't see the chat. Can't see the chat. Yes, Lou. Lavanya says, thank you for saying that. Right? Yes, I can't. 31 Adela, that's good, right? So here the clitellum. So we'll go back to the board. Happy to hear that you are still with me. If you have gone, if you have left the class, even I would not notice it. So let's go to this part. Well, we have a very short section, right? You can study the cell structure and function. So I have helped you to study the secondary growth part, and this one, so we are in annelid, check this annelids, people. Clitellum, parapodia, setae, and suckers are found in some members, found in some. So this is important, people. Clitellum, parapodia present in some. Now these things are now very important, people, in some. So if they might ask, clitellum present in all, right? Then you have to say it's wrong. Clitellum not present in all, in some. Clitellum secretes cocoon, which is sac, where the eggs are developed and fertilized. Parapodia are used for locomotion and respiration. That's the important point. Locomotion and respiration, both setae are present for locomotion, suckers for locomotion. Suckers present in some members. Now, something like people in leech, you can see that they have suckers. They have suckers. So that not only, now this one is what do you call, typical one. It's not an earthworm, it's not a leech, it's not a Bristol, probably a Bristol worm, right? So they have, so excretion, people, excretion, they have the nephridia, but it's not mentioned in the resource book. Later you will learn it in the excretion lesson, right? Now we move people to mollusca, right? Moving to mollusca, mollusks. Now you learn mollusks. You learn mollusks. Look at this mollusca, right? Mollusca. Now mollusks, the special feature is they have, people, pallium. Pallium is a special epithelium. And then the mollusks, they have visceral mass, foot present, mantle cavity present. This is pallium. Palliang nevi. Pallium, Pallila mariang, Pallium. This is pallium is a special epithelium, right? It's a special epithelium. Pallium is continuing like this. So this is the mental cavity. So this is the shell. This is shell secreted by the. This is a shell secreted by the mantle. Shell secreted by mantle. This is the internal organ containing part. What do you call that? Visceral mass. Ne? This is visceral mass. Visceral mass. And then people, they have this GI tract, the stomach, those are present here. And then it's open, GI tract open into the mental cavity, right? They have people, we said, this is the gill present, and then this is actually sending blood into the heart. Heart pumps blood into the cavity. Ne? So we learn this is receiving blood from this cavity. It's something like this. Ne? So this is a gill. The what do you call this blood, or what do you call the hemocoel? Ne? Right, the hemocoel goes into the gill, get oxygenated, transported into the heart. From the heart, it's going to flow into the hemocoel. So that's an open circulation. They have open circulation. And then people around the heart, gonads, no need. Then they have people, these excretory structures, nephridia. Excretory structures present nephridia. Now, this cavity inside the body is the hemocoel. This cavity is the hemocoel, right? So these organisms are having special chitin containing sharp tongue present. Ne? What do you call people? Radula. Ne? For the rasping organ is the radula. Then some members are having eyes, different types of eyes. Foot present. This is the foot. Right? Muscular foot present. So the body, if you take people, foot, visceral mass, and the pallium. Foot, visceral mass, and pallium. Pallium and visceral mass. Visceral mass and the foot. Visceral mass and foot present. Right? So these are the main features. Shall we check, people? The mollusca details. Phylum mollusca. Characteristic features of each example not necessary. So oysters, clams, slugs, snails, octopus, squids, chitons, and tusk shells. A lot of examples present. Ne? Okay, we'll leave this tusk shell. What are the features, people? Majority are marine. Some inhabit freshwater and land. Some are bilateral symmetry, and few are asymmetrical. So asymmetrical, especially people, garden snail and all, because of the spirally coiled shell, they are asymmetrical. They are soft-bodied and non-segmented. Calcareous shell present. Ne? Body divided into three parts. That's important, lamai. Muscular foot, visceral mass, and mental. Right, mental. Mental is the pallium, ne? Pallium or the mental. I think they have mentioned mental to secrete the shell. Shell could be internal or external. You know, some members are having external shell. Ne? Now, for an example, garden snail. Some are having internal squid. Right? Some don't have a shell. Octopus. Right? Many mollusks possess radula. K radula. A minutely toothed the chitinous ribbon-like tongue. Ne? Most mollusks have separated sexes. That means males and females present, but that doesn't mean most. Look at this word, people. Most mollusks have separate sexes. That means all mollusks are not having separate sexes, right? Gas exchange occurs through gills located in the mantle cavity. Excretion through the metanephridia. Nephridia, people, right? Metanephridia. Nephridia or the metanephridia. All right. All right. We have just 2 more phyla to go. 2 more phyla to go. Ne? Arthropoda and Echinodermata. Arthropoda and Echinodermata. Right? What's the time now? 8:57, 9 o'clock. 9 o'clock. Ne? Right? Arthropoda and Echinodermata. Right? Arthropoda and Echinodermata. Right? So, yes, I'll then give you the chance to go through the Arthropoda and Echinodermata. We took 7:30, 8:30, 9 o'clock, 1.5 hours. Ne? I think I have to keep my promises, right? So I'll disappear now, right? Leaving you to, right, have good night sleep. Now, so you have a job to do. Go through the cell structure function part, and then you have 2 more phyla to finish. Ardalamai, can you do that for me? Can you do that for me? Yes or no? Can you do that for me? Come on, reply me, right? So 2 more phyla are there. Yes, you can do ne. Small part ne. You can do that. And then people, I want you to finish the paper. I want you to finish the paper, right? And then I'll meet you people on Saturday, right? We'll discuss this paper on coming Wednesday. So complete the paper, submit it, and we'll meet once again. So thank you very much for joining with the class. Vanakkam, Aivon, and Salam Alaikum. I'll see you all soon in the weekend class.